You'll be modeling one of the 5 processes. So on this table we have diffusion. So if you come here, you'll be modeling diffusion. This is facilitated diffusion with channel proteins. This is osmosis with water. So you need to show hypotonic, isotonic, hypertonic on this table. And then this is endocytosis and exocytosis, and then that one's active transport. So you'll pick your table, and you'll spend about 10 minutes making your model. So you'll draw on the table with chalk markers, you'll add different items to show your modeling, and then you need to write up the step-by-step process of how it happens and what you're doing with the different pieces, what they're representing. Once you're done with that, and I call time, then we're going to rotate. So then you'll rotate to the 4 other tables, you'll follow their instructions, what they wrote on the table, and then you'll fill out the sheet as you model the other 4 processes.
How we did that model of like the cell membrane, could we like draw and then put like that cell membrane around the inner layer of it, and then put like the osmosis, like how it does it?
And so you're showing your 3 scenarios, probably in 3 separate cells.
Yeah, so can we just draw these and do our other things?
Yeah, but I would show the cell membrane like he was saying, and then you should show the aquaporin channels, because that's actually how it goes in and out. Yeah. So you could, are you going to show your water with those?
Yeah.
And then what are you going to show your salts with?
Like a different colored clay.
You could, yeah. Since the water's the stuff that's moving, you could do that out of clay, and then you could do your salts out of marker, since that's not moving.
Can we use this?
Yeah. Yeah, that's now it's drawn. All right.
Should I just do these then on like kind of like that side? Yeah.
That this is isotonic.
Yeah.
Oh yeah, so we.
So put
It's looking good.
use this. That's good.
And then you can write out the directions for each one.
Yeah.
So we're going to spend like 5 minutes at each one. This is endocytosis when it's bringing it in, and then that's exocytosis where it's pushing it out. So he kind of has it set up here where I think what he did was you can move these little guys. So this is the stuff that it's going to shoot out. It's too big to go through like a channel. And then it becomes like part of the membrane, open, and it becomes part of it, and then it shoots the stuff out. So that's that one. This one's the opposite. So this is isotonic when they're the same. The salts have the same concentration, so the water is moving in and out. This is the water. So the water stays the same. The cell stays the same size. This one, there's more salt in here than out here. The green is the salt. So the water is going to move in to try to dilute that salt to make it not so salty, and the cell's going to puff up. And then this one, it's saltier outside. So the water is going to move out to try to make it more salty inside, and the cell's going to shrivel. So that one's the cell's going to swell. That one will stay the same.
Okay.
Taking stuff from inside the cell and using it outside?
This is inside the cell.
Oh.
Inside the cell.
Outside the cell.
So it's bringing stuff inside the cell. With no energy. With energy.
Why does it need energy?
Because it's going from low